will not come into the picture. It is your problem. If you purchase a fish from the market, live or dead, you do and explain, you do anatomy or anything, yes. But if you give any drug to the fish, yes, CPCC comes. Okay. The next one, animals in the lowest first in the phylogenetic scale, based on the sentience, which may scientifically give valid results. You have to use. That means try to use the lower scale of the animals so that you will also not suffer. Animals will not also suffer. Okay. And alternative not involving animal testing should be given due and full consideration and sound justification provided if alternative and available not used. Because of the CPCC become very active, now the cell culture become very active. One industry leads to the other. Because of the ethics issue, now actually the robust science is, much of the discoveries are done in even cell line studies. Okay. So this is the second principle. The third principle is proper use of animals in experiments, avoiding the pain and suffering. Okay, pain and suffering of the animal. Many of the times we use the pie-edema model, carcinogen induced. Okay, and till you inject the animal, it will be keeping the leg on the ground. Then after that, it will like the elephant, it will keep, right? Because of pain and suffering. Okay, for pharmacy students, one question. Any compound I am studying, going to study the anti-inflammatory effect, X or Y or Z. Okay. I want to relieve the pain. How I can relieve the pain? But it should not relieve the swelling. Okay. It can be an analgesic, but it should not be an. What is that? It cannot be an anti-inflammatory. Is there any drug? Correct. Narcotics. But how many times people has used the narcotics and reported in the paper? And even when you induce a diabetes, 50% of the animal, when they induce more than 400 milligram, it will die. How many papers published in the scientific literature they have reported? None. Nobody has reported. And actually, animal experiment in a group, you need how many animals? Anybody can tell? For any experiment, when you do a study in a group, how many animals minimum you need? 6. Who told 6? How that 6 number came?
So
any experiment you need only 3 data. Any experiment you need to do it in, any experiment you have to do it in how many times? Triplicate. Even if you weigh a chemical, 5 milligram, 1 time, 2 time, 3 time, you have to weigh and confirm. And you have to, every time you have to measure 5.01, 4.99, 5 average. That is the weight. But because of fastness, we are nowadays only one time we are weighing. Okay. So what I mean to is only triplicate, only 3 data is sufficient to obtain the standard error. That is sufficient. But since the biological system will have variation, you have taken 50% chance. So if pass and fail, how much percentage of chance? 50%. So 3 plus 3 6. Right. And 6 animals are given. Then you have to report only 3 data, not 6. But people have started reporting 6 data. Then 10 data, 20 data. Okay. Here you go. So you have to use the animals with minimum pain and suffering wherever it is feasible. Okay. If animal is not, the experiment is not conducted, you have to suffer. So minimum pain and suffering. Then principle number 4. Person engaged in animal studies have the moral responsibility for the welfare of the animals after their use in experiment. Investigators are responsible for the aftercare and rehabilitation. After experimentation may be permitted after the experimentation. This is where the rehabilitation comes. Okay, that is the 4th R which I am going to explain. 4th R is reuse or rehabilitation. Okay. So after the experiment is over, you have to rehabilitate. Right. After the marriage, if divorce happens, you have to give the alimony. Right. Same like that. If you don't sacrifice the animal, then you have to maintain the animal till its normal death. So instead of maintaining, you can sacrifice them. I will tell one beautiful story. When I was doing my internship at Chennai Veterinary College, I don't know, that time I don't know, big car. It was a big car, I don't know the brand. Okay. They came and they brought some animal, which breed I forgot. Family came, mother, father and one son, daughter, 4 people. The animal is very healthy and it's very awesome. Then they came at the casualty at night 9 o'clock. My professor was there and we were doing internship. And sir, what is the problem? What is the health problem? No health problem, animal is okay. Sir, we want to sacrifice, euthanize. We ask him, why you want to euthanize? It is very healthy. No sir, we want to euthanize. It is not allowed because you have to suffer from some disease, then only it can be euthanized. Uncurable disease, incurable disease, it has to suffer, then only. Then he told, sir, we have maintained that dog like one of our family members. We have taken utmost care, each and every care. Now, if the animal, we have to go to Malaysia or Singapore, something they told. This happened in 1995. Okay, they had to go, but they could not able to take the pet along with them. But they don't want to give to somebody's hands. Simple reason. They don't want the animal feel that after going there, these people, they have left me in lurch. Okay. Because of that pain, and also they don't want to bear the pain after they leave, what happened to the animal. Neither the animal nor the owner, they want to suffer. Okay. Then my professor told me, see, I am a professor of surgery. Okay. I will take care of the animal like anything, pet animal. No, sir. Except the love and care we have given, we don't mean that anybody can give. So this is our request. Then we have taken the undertaking and we have to do the duty. Okay. So after experimentation, it's also regard is very, very important. So why I am telling this? Instead of maintaining that animal for another 10-15 years, you also suffer, the animal will also suffer. So try to utilize the maximum possible data collected from the animal and give you a goodbye. Okay. So animals only can be euthanized in these conditions. When the animal is paralyzed and is not able to perform its natural function. If you collect the organs, then done with. Most of the time, why we don't do, suppose example, if you want to study the interactions of two drugs or synergistic effect, directly we can go to the human trial drug, because 2016 there was a gazette notification saying that for academic purpose you don't need of CDRI, any CD scope permission. Directly if the drug's action is known for any new use, okay, what is that? It is called as repositioning of drugs. Okay, then like that if you want to do it, you can directly do it. No clinical data is asked, need not be asked. Okay, and like that you can do. And if you collect any organ, then you have to sacrifice. You have to sacrifice. So during the course of experimental procedure, the animal has been left with a severe pain. Then you can, you can sacrifice. In situations where non-termination of the animal experimented upon would be life-threatening to animals, suppose it becomes anxiety, then the animal handling is very difficult, no? So in such cases, cost and aftercare, rehabilitation of post-experimental art will be part of research cost and should be scaled per animal in positive correlation. And none of the funding agency in India has sanctioned even 1 rupee so far for rehabilitation. So don't expect the rehabilitation government will give funding. Next, principle number 5: the living conditions of animals should be appropriate for the species and contribute to the health. Okay, and the housing, feeding, and care of all animals used for biomedical purpose must be directed by a veterinarian or other scientist in a relevant discipline who is trained and experienced in the proper care of handling use of species. Circumstances, veterinary care should be provided. And one more thing, all the animal experiment you are carrying out under the veterinary supervision. This is very, very important when you present in international conferences. Don't tell that I am the one who conducted this experiment. Any experiment you are conducted is under the veterinary supervision. And other than a veterinarian, nobody is eligible to give an injection or a tablet or a drug to the animal. You are not authorized even as an MPharm or PhD students. Now the rule is coming. Okay, these many days it was less, and now Veterinary Council of India is framing the guidelines. It is under, that is why in IAEC one of the members is a veterinarian. Even in your IAEC also one veterinarian is there. Okay, without his knowledge, now that's why he is sitting in the protocol approval. No, he need not present. He is not presenting. Going to present in the word very nicely. It is used supervision. Supervision means it can be online mode also. Okay, that means it has gone to the brain. Whether it is stored or not, we are not bothered. Okay, so in the ethics committee, when the protocol is reviewed, veterinarian will be seeing whether that protocol will cause any pain or anything. He will suggest. Once he has signed in the minutes of meeting, then it is approved. But beware, any surgical procedures like wound healing or something, such studies when you conduct, you write in the paper side, conducted under the supervision of veterinarian. That is very important. Okay, because one of the international conference, one of the Indian, he has presented the, what is that, one of the oral talk. During that time, one of the international participants raised the question, and once he told that it was not under the veterinary supervision, immediately they told him to get down from the stadium. Very important. Okay, this is one of the basic ethics issue. And this is now 5 P's are over. Now 3 R's. 3 R's. First one is the replace. Whenever you are doing the experiment, first thing what this order should not be changed. Order is very, very important. First is replace. First is not refinement. First is replace the work, your research work with the animals, without animals whenever possible. The best alternative is human. For whom we are going to develop the drugs? Is it for animal treatment or plant treatment? Human. You are the best alternative. Okay. Now you tell me, can any drug directly we can test it in a human?
No.
What is phase 0? Anybody from beginning? What is called phase 0? Human microdosing. Do we use animals for human microdosing even though it is a new compound? That is the question, answer of my question. Human microdosing, because we don't have AMS facility in the world. We have only 3 instruments in the world. It is going to come. And one more secret I will tell you. World over, who decides what type of research has to be carried out? Do you know? Only 1 or 2 persons they decide what type of experiment you have to do it. What type of research you have to do it. Do you know who? Equipment manufacturers and chemical suppliers. Equipment manufacturers and chemical suppliers, they are the one who decides what type of research you have to carry out. And what type of disease has to be diagnosed and what treatment has to be given, that is also decided by the point of diagnostic devices. Before you don't have a glucometer, you never know there is a diabetes. Before you know SpO2, you don't know what is SpO2. Before you don't know how to use the blood pressure equipment, you don't know what is hypertension. They pumped your heart so that they will pump the money. Okay, anyhow, it is life for many. Okay, because everybody has to take a business, get a business. Okay, do you know which is the number 1 poison in the world? Anybody knows? Number 1 poison in the world.
Number 1 poison.
Number 1 poison. And who invented?
Number 1 poison.
I will tell you. Sir, who? Sir.
Prakasharan.
Number 1 poison.
Till 1960s, many of us we don't know many of the diseases. 1960, one of the, anybody can tell which is the world number 1 university in the world? Anybody can Google also. Top 3 universities, can you tell? MIT is number 1. Harvard, very good. Oxford, these 3. Okay. And now I will Google it. Maybe I won't be speaking the mic. Just watch it.
Number 1 poison.
Number 1 poison.
Sure.
Don't make noise. Just silently watch.
Sugar.
Okay. Which journal it has been published? And it has been revealed by Java Internal Medicine in 2020. No, not 21. 2017. It has been found. Oil is not the culprit.
Sugar.
And one of the scientists, he did not agree for this. Shunt out from the university. Okay. Only one. Remaining people paid. Okay. This 1967, when article came, they blamed the fat, not the sugar. And sugar is dash times addictive than cocaine. You fill it whatever the number you want. Sugar is addicted dash times than cocaine. You can Google it and find out. You will get shocked. And this is the one we add in the milk and give the baby.
Yes.
Right? This is the culprit for all the diseases. Okay, anyhow. If this is not there, then we don't have pharmacy also. Don't worry.
Sir, what about jaggery?
Minimize, sir. Minimize.
Jaggery, very good.
Don't close.
Who has asked me the question? Madam.
Jaggery is good.
Madam, jaggery, it is a unpurified form and it has little bit of fiber and it is in natural form. The problem is not the sugar, it is the sucrose. Okay, sucrose we don't, in our body we don't have the sucrase enzyme, so that we cannot cleave into glucose. In that, the major portion is fructose. Fructose is not an essential nutrient in the body. Glucose is essential nutrient. This, especially in the soft drinks, we use the fructose syrup. That fructose, it accumulates in the pathway. In the pentose pathway, if you see, the fructose will come from the one side and other side it will come the fatty acids. Both combined together it forms the triglyceride. Okay, triglyceride is the one which forms the adipose tissue. Okay, once this what is the triglyceride deposits is not only adipose tissue, wherever the membrane is, it go and block. So no communication. Get the disease. Be happy. Make money. Okay. So now
Doctor.
where is the rupees? I still have pricing. Yeah, this is rupees. Okay, first one is the replace. We already made rupees, we made cell culture and we are making money. Okay, replace with work with animals and many of the clinical trial companies are coming and India is guinea pig of the world. Okay, most of the, because well educated, less paid. Okay, many of the clinical research companies are there, one PHRI is also leading. So replace the animals wherever it is possible with the inanimate, invertebrates or those models. And reduce, and second one is the reduce. You can't avoid the investment. You have to do experiment. Then try to reduce the number of animals. The best example is like acute toxicity study, where you will be given only 3 animals for dose range finding studies. And earlier we used to do to find LD50. Okay, we have to kill ourselves 50%. Okay, those many number of animals. But now you need only 3 animals, 1 animal, you dose it, step up or step down. Whatever the way you do, you find it, or limit dose study you can find it. Okay. And even reduce the animals. Suppose you have to measure some of the tumor development. Since if you have 3 animals, you can induce the tumor. You know normal animal, already taken the X-ray. You induce the tumor, take the X-ray. If it is normal, then that also not needed. If it is ascites, that also not needed. You need to wait only take the weight and volume. Okay. Then after that, give the drug. And the same animal you can do it. You need no need of different groups. So you can reduce the number of animals also. Okay. Then refinement. Refinement is nothing but whatever you do. As example, anti-inflammatory I told no, you can give some of the narcotics and you can study. That's called refinement, to give minimal pain and suffering to the animal. So now the suffering is when we detect. For many of the people they will be thinking, sir, we are suffering from. Okay. This is the replacement strategy. I don't go in detail. And this is the guidelines. Okay. The first one is the compendium of CPCCA. Anybody want to become a CPCCA nominee? That is the Bible. Okay. You read the Bible and you can preach in the committee whatever you want. Okay. So that has all the guidelines is there. Till 2020 or 19 they have updated and it is there, everything. Just go there, just put Ctrl F, find the word and you can read whatever you want. Okay. And role and responsibility of nominees of CPCCA, that is given. Only the nominees they need to read. Guidelines of our constitution and reconstitution of institutional ethics committee, usually 5 members from the internal, 4 members from outside, totally 9 members, minimum 6 members should be there. Okay. Guidelines for the use and rehabilitation of animals, then amendment for the protocol CPCCA for the poultry, and finally they have fished out the fish. Okay. So till 21 it was geography was not under the CPCCA, but in abroad it was there. Any invertebrates it is there in that. So this is the one of the guidelines, CPCCA guidelines for the amendment facility, which tells that all the guidelines how to. So you can see the first paragraph. Experiments shall be performed in every case by or under the supervision of a person duly qualified. That behalf is degree or diploma holders in veterinary science, medicine or laboratory science or university interned for the purpose and under the responsibility of the person perform in the experiment. It's very clearly given. Okay. Like that it is there 27 page, you read afterwards. I am smart now. So this is the composition. You can see the top 5. Yeah, actually 5. The second one is 2. It's a duplicate. Huh? One biological scientist, 2 scientists from different biological discipline, 1 veterinarian and 1 scientist in charge of animal facility, and the chairman can be anybody. And suppose if the administrative head, if he is a BCom or a BE engineer, he can be also a chairperson extra. So 6 persons. Okay. So next is the nominees. There will be 4 nominees. One is the main nominee, another the lead nominee. Whenever main nominee goes out of station, out of country, then the lead nominee will be coming and one meeting in a year. And there's sometimes there in compendium. This is how it will look like. This is the registration number valid up to actually day for yesterday my license is over. Okay, just like renewing the license, it is still pending. Okay, but annuals is not closed, it is ready. Okay, so this is like biological scientist nominee like that. And this is the IAC has been designed for the following, and this is the form B. Many of you would have seen. Okay. This is the form B. What are some of the information they will be asking based on the 3R principle? Especially 3R principle, it comes under 8ABC, where you will find very difficult to do. It's rational for animal usage. 8A, why the animal usage is necessary? Why you can't replace the animals? First R. Second R, why the particular species is required? Second R is why the estimated number of animals required? Why you can't reduce the animal? Number 3, all similar experiment has been conducted, or any in vitro data is there, serial number 10. Okay, and anesthesia, they will be asking serial number 11. That is the 4th refinement. Okay, if you tell right no there, if you do an anti-inflammatory study, if I come with any narcotics, not for you, for the animal. Veterinarian can do it. If you tell that no, NSAIDs are not allowed, like that, if you write it, yes. Only non-narcotics can be used like that, you have to write it there. Okay, otherwise, that is why that form. And survival surgery. Survival surgery, definitely there will be a veterinarian, and whatever the hazardous or the serial number 15, whatever you use, you need the IBC clearance. Okay, this is the declaration that you will maintain all the records. This is the CPCC address, and if you have anything, you can contact them 10 to 5. Otherwise, also you contact them. Otherwise, what you can do, if you want to be smart, you message me, I will solve your issue. So, this is what animals are telling. We don't smoke. We don't drive. We don't wear makeup. We don't use paint. We don't drink alcohol. We don't drop bombs. We don't take drugs. Just because humans do it, why we should?
So sad.
2035, no animals will be experimented in Europe. And 2050, no animals will be tested in India. Okay. So because human microdosing, and another one more, this one also coming. What is that? Microchips. Okay. Now integrated microchips also coming. It is the first Nature paper which has been based on the lungs they have developed in 2016. First Nature paper it came. Okay, microfluidic devices. Okay, now that is also going well. So with this, my presentation is over. We can take up some questions. Otherwise, I can. Don't laugh.
Don't clap.
You can note down my email ID, but don't send email ID. You note down my mobile number, but don't call. Only WhatsApp.
WhatsApp, great, sir.
In my institute, I am called as WhatsApp scientist. Even as madam, no, next lab my student will be sitting. Okay, they will do some experiment. They will photo and send. Sir, this is sedimented. What I have to do? Do you know it is advantage to me? I will go to Google and ask what to do. Then immediately I will reply. This is has to do. Then I am a great scientist without no knowledge. Thank you.
Any questions?
Any questions? For all the problems, there is a solution. For all the questions, there is one answer.
Will WhatsApp questions, sir?
Yes. Madam, don't answer. We WhatsApp the questions, sir, because I am Google is my guru.
So this rehabilitation is applicable to small
Mike.
animals?
No, a very good question. Sir has asked a very nice question. Rehabilitation is not applicable to small animals. Mice and rats, but rabbit and guinea pig applicable. So, what you do, something you do it with some organs. That's all. So that will be. Otherwise, mice and rats it is not applicable. Rabbit and guinea pigs it is applicable, but many of the days you can reuse it. But not more than 3 experiments in a single animal. That is there in the guidelines. Any general question also you can ask, or you can WhatsApp. Because the first question whenever my PhD students or the dissertation students comes now, first you tell what you want to do. And the student which she has got Prime Minister Fellowship, when she joined with the UGC JRF, I asked her, why did you join for PhD? Sir, JRF they use 35,000. They will take dowry when marriage. I want to save money. And the girl who has went to Germany also, she told, sir, after PhD I want to do PDF. I told now only go for PDF. Going abroad, 2 years of experience in outside lab. Why you have to wait till you complete your PhD? In between you can go to abroad and do the fellowship. Very easiest thing. And how I communicate for 20 journals I will tell you. I told I will send my students, I will take some 5 minutes. Sir. I will tell my students, see here, Goa there is a conference. You will make some what is abstract and send. Immediately, our students within 30 minutes they will come with their abstract, sir. Go to the Goa and come back. Example I told. Okay. Then come now. Yes, sir. Now what to do? Communicate. 2 journals. Okay. How we will do for 20 journals? Very simple. From is my email ID. 2 is also my email ID to me only. There is one thing called as BCC. What is that BCC? Blank carbon copy. There if you put even 1000 email ID, it will look like it has gone to only single person. Okay, that is the smartness I have. 20 email IDs of the editors. Like that. Then start. Dear editor, whether male, female or transgender, no matters. Dear editor, because I don't want to spend time, comma. Please find enclosed research work title. That is actually I have enclosed only abstract, not research work. But it's a research work, no?
Yes.
Please find enclosed research work title so and so and so and so. Full stop. This is one of the important area which many of the scientists are digging on. Not digging on, researching on. Okay, working on. And this is the first report on blah 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 blah. Every report is first report only. Okay, like that. Then we are looking forward for possible publication in your esteemed journal. Your suggestions are highly, what is that? Highly solicited. Solicited. Okay, like that. Then send mail. We will wait for 48 hours. Don't open the mail after 48 hours. Blah 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 like that. At least out of 20, I would have got 6 to 7 responses from the editors. Many of the editors they will tell, you submit through the manuscript submission portal. That portal we know. This portal you don't know. Okay, 6-7 people they will what is that? They reply. Among that, whoever is the top impact factor. Simple. I have taken my abstract and told anybody is ready to take my paper. Whoever is giving the maximum rate, I will sell to them. Okay, what I will do, I will select the highest impact factor. Then in that format, we will start writing the paper. Till that time, we did not write the paper.
Great.
Okay, this is not the one simple technique, smart technique I have done. I have multiple techniques. And for your information, I will tell you, I am the highest paid employee of the entire CSIR setup, which has 49 laboratories. Right, my gross salary is 3.5 lakhs and take home is 2.37 lakhs after income tax deduction. And still I have 11 years of service. And I have been paid more than my director and DG. That is the smart. Because this is my 4th government job. Jump like a monkey. Not monkey. I ride on a monkey and jump. I did not do any experiment during my PhD. Till date, I did not do any experiment. My scientists are doing. Just I keep motivating them. Older scientists difficult to motivate. Younger scientists, no, he will get promotion. Come. And don't worry that Don't never think that your idea can be stolen by anybody. With the original idea, never it can be sold. Okay, original is original. Don't worry about that. And those who cannot produce original idea, they will patent it. Those who have original idea, they will make money out of it. That's all. Clear? So now what they have done, we have auctioned it, then the journal comes. Then we start writing the manuscript. Okay, we sent to one journal, which is having maximum impact factor. Do you know why? To get rejected with comments. Then we will make the bride ready. Then we will send engagement over, marriage over. Okay. So same three-page paper, one of the paper, it's called dose call paper, dose calculation between animal and human. Okay, if you put dose call CF there, it will come. It is only 3 page, one small software, only 24 hours we work. And even one of the anti-inflammatory work with probiotics, it's published in International Journal of Biological Macromolecules. 24 hours work, you know. That paper published in 8.7 impact factor. And dose calculation between the animal and human species, we have developed a software 3 page that is published in 4.8 impact factor. There is people to buy your commodity, but you don't know how to sell it. That is important. Like this rejection rate is very less, and you know who is your buyer. Accordingly, you can make it the journal format, and always see that at least 3 references you put from those journals. That is one of the secrets. That is bonus. Sorry for your time.
Please be seated.
Thank you. Please.
Sir, we are very thankful for your choiceful, factual talk on CPCSEA, also IAEC procedure and protocol. I'm sure that everyone who are present here have understood about the principal guidelines, IAEC, composition, everything without any difficulties. Generally, the last speech will be very boring, though it is very interesting because since morning they used to listen and they feel it is very boring. But I literally felt it was like just we began our workshop. Yes or no? It was like that. Just we began our workshop. It was like that, sir. So the entire credit goes to you, sir. We are very thankful.
Credit goes to KLE. Who invited you?
Definitely, sir. The KLE also to you for sparing your valuable time with us. Thank you very much, sir. Now I request Mrs. Karavati to present a memento as a token of respect to the sir.
Yeah, this memento is on all of you. On behalf of you, I will receive.
Thank you.